Hi, hello everyone. My name is Juan Deras. From Brazil. I'm going to be chairing this session this afternoon, and I welcome everyone. Just a quick question that for people that are online, we won't be taking any questions from you for this session. That's the last session of the day. And then when we finish all the presentations, please stick around for the ending ceremony that we're going to have. And I will pass the word to Dominik, who has 20 minutes for your presentation. Welcome.
Thank you, and hi everyone. Thank you for joining this session as well. Please bear with me. I know it's the final session. It's late in the afternoon, but we're almost at the finish line. My name is Dominik. I'm a lab animal veterinarian. I currently work at Columbia University as a clinical veterinarian, and I work with Professor Huw Golledge at my vet school in Vienna as well in the field of veterinary ethics. And I'm going to give you today a quick overview on the implementation status of the harm-benefit analysis in European policy documents and also in European legislation. Now, I don't know how many are familiar with the harm-benefit analysis in the research animal field in general, and therefore I'm going to give you a few background info in the beginning. The European Directive 2063 EU is more or less the cornerstone legislative framework in the EU, that regulates the care and use for animals used for scientific purposes. Within the directive, it is stated as well that a harm-benefit analysis should be performed or carried out during the project evaluation process. At the end of the analysis, the harm inflicted on the animals should be justified by the project's outcome or benefit. One of the goals of the directive was to implement coherent standards and therefore creating a level playing field within the research community in the EU or within Europe in general. And by creating those standards, you would achieve a level of harmonization. And that harmonization should lead obviously to high quality research, data that is reproducible, and then also animal welfare standards that will be standardized and implemented and upheld by the community. Therefore, the HBA is a really substantial tool in the research evaluation process. Now, it all sounds theoretically great. However, unfortunately, the HBA is quite a challenge because the question always comes up as to how do we apply the HBA. I'm just going to abbreviate it now. The HBA in a practical context during the evaluation process. One of the main issues is that there isn't any clear methodology that would really allow us to practically apply this analysis during the process. One might obviously assume that there are national guidelines or policy documents that would give us some idea on how to approach this problem. And that was one of the intentions of initiating this study, and also the goal here more or less to find out similarities and also differences of the HBA implementation status in national guidelines and documents. Now, just some info on the materials and methods that we used here. This is obviously like a European issue, so we looked at all 27 EU member states here. We've also included Switzerland, Norway, and the UK. Reason for that is that the regulations in those countries are relatively similar, or they align with the directive or the directive's content. For example, the UK's legislature is at the time, the UK was still an EU member state, so therefore it is more or less the directive's content. 3 steps were used. First of all, we were looking at the transposition of the directive's HBA requirement into national laws. I'm going to go into detail in a bit more in the following slides. Second, we did a straightforward literature survey just to identify some guidance documents that are currently out there. And then based on those findings, we performed a cross-national comparison of those guidance documents. For the transposition of the HBA requirement, we basically just looked at all of the national laws and looked at the wording that was in those national laws and directly compared it to the directive's wording. And we're trying to find out if there were any differences. Now, obviously, there are like a lot of European languages, so we were heavily relying on language, on translation software, and then also whenever it was possible on native speakers to help us with the correct translation here. Literature search, pretty straightforward. We used some predefined key words and then used all kinds of databases and search engines. the time frame was typically starting from the issuance of the directive in 2010 all the way up until July last year. And on top of that, we've also reached out to national committees and asked them directly whether the countries had any national guidelines. Those national committees are more or less advisory bodies to the competent authorities that are ultimately carrying out the HBA and then making a decision on the research project. For the cross-national analysis of the guidance documents, we looked again at the HBA requirement in the directive, and we've broken it down into 4 domains of the wording. We've identified a harm domain, a justificatory domain or justification of harm domain, an outcome benefit domain, and then an ethical considerations domain. All of these domains were taken directly out of the directive and of the HBA requirement, and those 4 domains constitute the HBA requirement. Now, what we wanted to find out is, first, did the guidance document implement each individual domain? And by implementation, we were looking for a definition of the domain in those documents. And then second, we wanted to know if it was operationalized, meaning was there a practical method in those guidance documents that would tell us how to practically apply this domain in the review process during the HBA. Looking at some of the results here, starting off with the transposition of the HBA requirement. On this table here, more or less, there's a lot of like hyphen and minus on here. Basically, wherever you have a minus, there were no differences in the national laws. However, we did identify 5 national laws that had some differences in their wording, namely in the national laws of Denmark, Finland, Germany, France, and Sweden. And then on top of that, we've also had one special case, and that was Austria. Austria is the only country or within the EU that has an additional regulation in place that is pertinent to the HBA specifically. So it requires project applicants to fill out a so-called criteria catalog as part of the application with the goal to objectify the HBA. For the literature search, as I mentioned, pretty straightforward. Unfortunately, using the databases that we had, like PubMed and all these like typical ones, weren't really helpful because these documents aren't really part of scientific publications. So we were heavily relying on Dr. Beaulieu here, and then also on the feedback that we received from the national committees. We've identified in total 7 guidance documents. Out of those 7, there was the EU document as the overarching document that was published by a European working group at the time of the issuance of the directive that kind of gave you some guidance on the project application evaluation, and the HBA in detail. And interestingly, what's noteworthy here is as well the feedback that we received from the national committees. Almost 75% came back to us, and the answers were mixed. Some of them were saying, well, no, we don't have one, but we're using the HBA EU, the European document, as our guidance. Some of them were saying, yes, we do have our own national guideline, and those countries actually aligned with the ones that we found during our search. And then others were saying, no, we don't have one, but we really want one. Can you please send us one? So it was really a sign for us as well that there's a need for some sort of guidance here. Now, as I mentioned in the beginning, we've tried to compare these guidance documents based on the 4 domains that we've established. In a first step, we wanted to identify the level of implementation. Is there a definition of, for example, the harm domain present? And then second, is there a form of operationalization in the guidance document? Does it tell me how to use or how to apply the harm domain? If a question was answered with yes, say, yes, it tells me what harm domain means, we gave a score of 1. If it also operationalized that domain, we gave an additional score. So per domain, we had a max score of 2. On this histogram here, we've basically compared now all 7 documents. And as you can see, starting off with the harm domain, that is the black colored column here, pretty consistent across all 7 documents. So yes, we did have consistent implementation and consistent operationalization. Now, after the harm domain, it gets all a little bit mixed here. Starting off with the outcome benefit domain, we do have at least some sort of implementation in those documents. However, when it comes to operationalizing already the outcome benefit domain, most documents are relatively silent here, and they don't really tell us on how to practically apply it, except for the Swiss document and the UK document that we've identified. And if we're looking at the justificatory domain, really the weighing part of harm and benefit, it is kind of all over the place. So we have one document that doesn't have it mentioned at all, the Belgian document. Others at least have a level of implementation in place. Some documents try to operationalize it, like the EU document. And then there's the Swiss document that really gives you a straightforward method on how to really weigh harm against outcome and benefit. And then lastly, the ethical considerations. As you can see, there's only one final guidance document, the Dutch one, that really attempts on addressing this domain. Now, in the next step, we've tried to illustrate individual guidance documents in a more precise or in a more individual way, so we can draw really one-to-one or one-to-two, one-to-three comparisons. So we've tried to visualize them in radar plots. The concept here is pretty much the same. This is the radar plot for the EU document, the EU guidance document. You have four corners. Starting on top, you have the harm domain, followed by the justification domain, outcome benefit at the bottom, and then ethical considerations at the last corner. We have, again, the same scoring mechanism here. So if we have a level of implementation, a score of 1 was given. If it was also practically applied, another additional point was given. Now we have also two squares here. It's a bit hard to tell, but there's an inner square that is highlighted in light blue here, and then an outer square that has a white background. The inner square more or less just represented the level of implementation, and the second outer square was the one that we had for the operational level. If you're looking at, for example, the harm domain, you can see that the HBA EU, the EU document, got a max score of 2. So we had really a level of implementation, a definition of the harm domain, and also told us how to practically apply it in the HBA. At the outcome benefit at the bottom, for example, we've only had a definition of the outcome benefit, but it didn't really tell us on how to practically apply it in the analysis. And then for the ethical considerations, for example, there wasn't really anything in there. So we were able to really create these radar plots for all guidance documents, and then really draw some clear comparisons and also some conclusions in the end. As an example, we have 4 documents up here. In the middle, you can see the Austrian and the French document. Both documents are relatively aligned with the coverage of the domains. Aligned also not only with each other, but also with the EU document on the left, on the far left. And then in comparison to that is the UK document on the far right that more or less is also relatively aligned with the EU document, but goes even further than the EU document when it comes to the outcome and benefit domain. This comparison also allowed us to really see which documents are the most comprehensive ones. The Dutch document here in the middle, for example, is the only document that really tries to address all 4 domains. Doesn't really fully achieve it for each of them, but at least it tries to address them. And then on the other end of the spectrum is the Belgian document that really only mentions the harm domain and the outcome benefit domain. And then again, in direct comparison, we were able to see that the Swiss document is the one that is really the most comprehensive one, at least in terms of 3 out of 4 domains for the harm domain, justificatory domain, and outcome benefit domain. But it really leaves out the ethical considerations of the HBA requirement, which is also fine given that the Swiss legislature is not really part of the EU directives content. And then just some final remarks here. We can already see that there is a discrepancy of the HBA implementation at the level of the national laws, given all these differences that we have in a handful of countries with their legislation. There is clearly a lack of guidance. We've only identified these 7 documents that give us some sort of idea on how to practically apply the HBA during the review process. And whatever we found is really far from being comprehensive. None of the documents is really there to fully tell us on how to practically use the HBA. So it might be even more conflicting than helpful at this point by using one of these documents. And just circling back as well to the directive's initial idea of creating a harmonization across European countries with the standards that are implemented, it is really questionable at this point to what extent, to what degree the harmonization is present, and to what degree the harmonization is really available. I do have up here a QR code if anyone's interested in publication. It's really just, I just scratched the surface here. There's a lot more detail in that paper as well. And with that, I'm happy to entertain some questions.
Thank you, Dominik. We have Bjorn here right in front.
Thank you. And thank you for a nice talk. It said that the devil is in the detail. And I sometimes think that a legislative text, I mean, the effect of that depends on how it's interpreted. And even in one country, the difference between different boards can be huge. So have you been thinking about actually going out and doing a survey to the ethical boards, or presenting a number of cases and asking, all right, so how would you evaluate this? What considerations would you take into account, etc.?
Yeah, that's a great comment. And we actually know of collaborators as well in Sweden that are doing these surveys with like the ethical review boards over there, and see at least how they are approaching the review process in a consistent way or not. Because as you mentioned, it is really different across the board on how they do that.
Have one up there.
Hello. Yeah, thanks. Sarah Dusel from the University of Hohenheim. So you showed in your scoring that some of the national legislative texts scored lower than the EU text. And I was wondering, I mean, the directive sets a common goal that everybody has to achieve. Does the European Commission file proceedings against any of the member states? Thank you.
Yeah, so the, let me just go back quickly. I guess you're referring to this one maybe. So this was really pertaining only to the guidance documents, which do not have legal character. However, I mentioned as well that we did find some differences in the national legislation as well. And for Germany, for example, we've also found some differences there. However, in that specific case in Germany, the German legislation mentions all 4 domains of the HBA requirement of the directive. However, it is not word by word written in the German law.
Yeah. Steve Wickens, UFAW. So you just published this. So what are you looking to do next with it? So presumably you're going to be going to certain countries and saying, Belgium, did you realize that you're really not doing well compared to even the basic EU standard? What are you going to do about it?
I think they know. In the next step, we are actually working right now on a HBA method that we're trying to put out there, and to be really, I didn't go into detail in the beginning about it, but there are currently 2 methods on how to approach the HBA. One of them is the metric approach, and the other one's a discourse model. Basically, with a metric method, you are really following a strict step-by-step algorithm that will tell you if a project will be approved or not. With the discourse model, which is more commonly used at this point among the reviewing bodies or ethics committees, is more or less where you just discuss the topic and then come to a final conclusion. And we're trying at this point, based on the results here, that there's really no clear guidance. First of all, propose a mix of both methods, see if that would work, and then also come up with a guideline itself that should hopefully be helpful.
Joe.
So, Joe Garner, Stanford, obnoxious question asker. I've always really struggled with the whole concept of harm benefit analysis, because it would seem to, everybody draws it as like a seesaw, right? Which implies that there is a common currency to harm and a common currency to benefit. And I fundamentally don't see how that's true, point number one. And point number two is that harms are definite, right? We know there are going to be these many animals, we're going to do this to them. Those harms are definite. The outcomes are always probabilistic, right? So, you know, if we think about cancer, for instance, if you actually sit down and you plug in current reproducibility of cancer studies, which is about 10%, if you plug in current translation rates of cancer studies, which is about 3%, you would need, on average, to initiate, sorry, guess what? I've done the math on this. Like 950 new products to get a single cancer drug to market. I don't see how that probabilistic component is ever being incorporated into these models if anybody is ever approving a cancer study.
Right.
You see what I'm saying?
Yeah.
And so to me, the whole thing seems horribly disingenuous because we're going through this exercise and then we're just going, well, we're going to approve it anyway.
Yeah.
So that is my incredibly opinionated statement. And I was wondering if you would tell me why I'm wrong.
Okay, sure. So I don't think you're completely wrong. Just, you know, starting with the issue of the wrong or like not having clear definitions is really important because there's also like a clear difference between, as you mentioned, an outcome and a benefit. Outcome in the end, you know, is always going to be your result, your data. However, your benefit can be something different, right? Your benefit will be the positive influence on the animal, humans, or the environment. And then weighing those two currencies, like in the past, like this talk also had the title of like, how do we weigh apples and oranges? It really is not clear, right? It is a challenge. And I think we've also shown here that like having all these different guidance documents out here with different opinions is just really not straightforward. And trying to find a common sense here is really going to be crucial. And that's what we're trying to attempt as well in the next step.
All right. Thank you, Dominik.
Thanks.
So our next speaker is Michael. Feel free to use the word.
Okay, good afternoon, everybody, and welcome to my talk entitled Connecting Biologging and Wild Animal Welfare. So as we heard yesterday and today, the field of animal welfare is currently expanding from the study of captive animals to the study of wild animals here living under natural conditions. And of course, this complicates the way we can assess the welfare of these wild animals. First, because typically these animals can move freely in their environment. And of course, this complicates their direct monitoring to assess their welfare. Second, they experience variable conditions that they cannot control as opposed to animals in captivity. And finally, there's also a great taxonomic diversity in the wild, which might require you to have a broader taxonomic knowledge, at least broader than birds and mammals as typically considered in captivity. Recently, my colleagues and I at Wild Animal Initiative have listed all the indicators that could be used to assess the welfare of animals, and we assessed their suitability to use them in the wild. And you can see that there is a great diversity of indicators ranging from physical measures such as body condition or physiological indicators to behavioral measures. But despite this diversity, most of these indicators could be measured by using a single methodology, which is biologging. Here, I refer to biologging as being the use of electronic devices attached to unconstrained animals to store and or transmit physical and or biological data. So I try to be as complete as possible here. But it may sound complicated, but I guess you already know many of these electronic devices, such as accelerometers to measure body movements, geolocators to measure movements over large geographical scales, acoustic recorders to measure vocalizations, or proximity loggers to assess inter-individual interactions, as we saw before, for instance, with the lamps and the hues. The interesting thing here with biologging is that these electronic devices will allow you not only to measure these potential welfare indicators that you can see here, but they will also allow you to measure at the same time the environmental conditions that animals are experiencing. Indeed, most of these indicators also include some sensors that can measure environmental conditions such as temperature, light, or humidity. And of course, this combination between welfare monitoring on one side and environmental monitoring on the other side makes biologging a potential great tool to assess the welfare of wild animals. Because, of course, it can compensate for the challenges I was mentioning before in terms of free movement in the wild and in terms of uncontrolled conditions, so conditions that you cannot control, but at least that you can monitor with these devices. So this sounds great, but is it something that people have already started to use to assess the welfare of animals in general and the welfare of wild animals more specifically? So to answer this question, I reviewed the biologging and the animal welfare literature, and I examined the overlap between these two fields. You can see that I focused on the research articles that were published in the last decade to reflect the most current trend. And I targeted my review towards these journals that I assumed were representative of their respective field. In biologging studies, I examined whether they included some welfare-related terms such as sentience, emotions, or affective states, while in animal welfare studies, I examined whether they included biologging-related terms and if they included biologging approaches. Here you can see the result of this review. You can see that very few biologging studies include welfare-related terms. And this very small proportion is mirrored by a very small proportion of welfare studies using biologging approaches. What it means is that at the moment, animal welfare is very rarely assessed through the use of biologging, and I'm glad that there was a talk before that used this methodology. I guess you were representative of this review, like 7% of the studies, of the welfare studies use biologging approaches. The situation is even worse now if you consider wild animals, as biologging studies including welfare-related terms were conducted in only half of them were conducted in wild animals. And you can see that for welfare studies using biologging approaches, they were very rarely conducted in wild animals. And this means that there are obstacles, apparently, that prevent the use of biologging to assess wild animal welfare. To identify these obstacles, I evaluated biologging against the criteria that are currently recommended to assess animal welfare. You can see here that there are three criteria: correctness, usefulness, feasibility. If you are not familiar with these criteria, please have a look at the reference below. And you can further break down these criteria into several sub-criteria. For the sake of time today, I will not talk about each of these criteria, but I will talk only about two of these criteria that might be considered as potential obstacles for the use of biologging to assess wild animal welfare. You can see that these two sub-criteria are validity and disturbance. Let's start with disturbance. So I guess most of you already know that biologging implies the capture, the constraint, the equipment of animals with data loggers, and the release of animals with data loggers into their natural environment. And of course, each of these steps can generate high levels of stress. There are, of course, some techniques to minimize this stress, but you cannot completely eliminate the stress generated by these different steps. In addition to that, the devices themselves or the physical characteristics of the devices themselves can generate some stress. For instance, the color of the devices has been shown to generate some stress in some species. But more importantly, other physical characteristics such as weight, size, and how streamlined the devices are can strongly reduce the physical capacity of animals in their environment. And this is important because, of course, these animals cannot find their food as efficiently as if they were without any devices. And of course, also they cannot escape predators as efficiently. These side effects are so important that they have become actually the main focus of biologging studies considering welfare aspect. You can see that more than half of these studies focus on these side effects, as I found in this literature review. Quite paradoxically, welfare studies using biologging approaches do not really focus on these side effects and tend to ignore these effects and focus on other welfare-related topics. These side effects also will limit the range of species on which you can use biologging approaches to assess animal welfare. As for a given size of device, larger species, of course, are expected to be less impacted than small species. And this is exactly what I found. You can see that biologging studies considering welfare aspects and welfare studies using biologging approaches are mostly conducted on large or very large species, while small species are almost absent. But there is some hope. I think this is what you have to remember today from my talk. There is some hope. In the last decades, the size of these devices has consistently decreased years after years, so that biologging approaches are now not limited to these quite large species that you can see here, but you can now apply such approaches to smaller species such as bats, songbirds, or frogs, and even tinier species such as insects. You can see here on the graph on the left side that the size of the devices has also decreased when they are used on insects in the last decades. So I hope in the next years that small species and even insects for which the sentience is not necessarily completely clear will appear in these studies combining animal welfare and biologging. Let's switch now to the second obstacle that I identified, which is validity. So here I define validity as the ability of an indicator to reflect the affective state of an individual or an animal in that case, as opposed to any other state. To make it more simple, the question here is to know whether these indicators that I showed you before really reflect the affective state of or the welfare of animals in terms of affective states. Recently, our collaborator, Heather Browning, has suggested a validation procedure made of 3 consecutive steps. The first step is that you have to formulate assumptions about the valence value of the affective states experienced by animals under certain conditions. In other words, you have to assume whether certain conditions are pleasant or unpleasant when animals are exposed to them. Second, you have to measure whether being exposed to such conditions is associated with a physiological change or a behavioral change. And here, this is where you would have to choose one of these indicators and choose a relevant device or data logger to measure this indicator. For instance, you might want to measure inactivity with accelerometers. And finally, you would have to examine how consistent these physiological or behavioral changes are across a variety of conditions for which you assume the same valence. To make it more simple, I added this graph here. Here you can see that I considered 7 conditions for which I assume a negative valence. I can be quite confident that being injured, being exposed to predators, being exposed to noise or human activities is quite unpleasant to animals. So it should result in a negative welfare. Now, if I measure the movement of this deer with an accelerometer, and if all these conditions lead to the same change in terms of movement, then I can assume that this movement change is indeed a valid indicator of poor welfare, because it reflects the only characteristic that all these conditions have, a negative valence. So now that I have validated this movement change as being an indicator of poor welfare, I can use it to further validate other indicators. This is what we call a triangulation procedure. So I can see, for instance, if my movement change as assessed by accelerometers correlate with social interactions as assessed with proximity loggers, or a change in vocalizations as assessed by acoustic recorders. And of course, in that situation, multi-sensor biologgers are highly useful, as they would allow you to measure all these measurements at the same time and see how they correlate under given conditions. Okay, to conclude, biologging could strongly contribute to solve many of the challenges related to the study of wild animal welfare, but there is at the moment only a very small overlap between the fields of biologging and animal welfare, which is presumably due to the limitations that biologging has in terms of disturbance and in terms of validity. But as I said, there is some hope. There are solutions that exist to enhance, to reduce these limitations and to enhance biologging approaches. And I hope that these solutions will encourage you to use biologging in the future to assess the welfare of wild animals. And with that, I would like to thank you for your attention. And if you have any questions, I will be happy to answer them. Thank you.
Thanks, Michael. We have one question right here in the middle.
Thank you. Thanks for a really interesting talk. I was wondering if you could comment on the risk that comes with putting biologgers on wild animals, in the sense that the biologger itself could compromise the animal's welfare, and as compared to domestic or captive animals, where you can actually continue to observe them after it's been put on, and you can assess that risk very clearly. And if any of the studies that you reviewed address that in any way.
Yeah, so there are many studies, actually many reviews on this topic in the wild. Not only animal welfare scientists can use biologging, but actually this is a technique that animal ecologists in general use. And here, as I said, so they are interested in welfare because of these side effects. You can measure actually these side effects, for instance, in the wild as opposed to captive animals, by using either different data loggers that you expect to affect the welfare of animals more or less, like twice the size, twice the like a streamlined data logger, not a streamlined data logger, and you can compare both. Or you can also measure indirect markers. For instance, I worked on penguins. That was my first slide. You can measure how long the foraging trips are. So you don't need a data logger for that. So you can use a real control group without any data loggers. You see, you look at how long the foraging trips are, and you compare that with the group with data loggers. You can also look at the reproductive performance, for instance, also between these two groups.
Thank you.
Have Mark over there.
Hi, Mark Russo, Harper Adams University in the UK. One way to overcome the disturbance would be to look at non-invasive approaches. Did you pick up any publications using drones? I know drones are being used to monitor wildlife, and I think with machine learning and some species, possibly with individual differences in their markings, you might be able to monitor the animals from drones. So is anyone using that approach? Do you think there's any potential in using drones?
Yeah, so drones are also being used by animal ecologists a lot, and they have also measured some side effects on the welfare of animals. So you have to be very careful of that, actually, with drones as well. I forgot the first part of your question. But yeah, so I can see the potential of using both, actually. I'm insisting on biologging today, but there are other methodologies, of course. You can combine both and use the least disturbing method.
Yep.
Hi there, Aileen McClellan, University of Ottawa. I was wondering, when you're looking at overlap between the fields, in your list of welfare relevant terms within the biologging studies, I think stress was not included in that. And so I think I'd like to confirm that I didn't just miss it. But if that's the case, I'm wondering why you would choose to do that, because it seems so relevant to affective states that those researchers might be considering.
Yeah. So it was a voluntary decision not to include stress. Actually, I did that for another review that I did last year. So it's correlated. The more articles refer to welfare, the more they refer to stress as well. But stress is so much loosely used most of the time that I decided to exclude it because it's more like a confounding factor than bringing extra information. So I think it's better to exclude it. And we have also to be careful that physiological stress is not necessarily an emotional stress. So I think that's why we have to be careful when we want to include that. But it was also a very important question I had at the beginning. Should I include that or not? And I saw more disadvantages than advantages.
Steve Wickens, UFAW, it's sort of related. So in terms of the overall number of papers that were published on wild animal welfare, did you look at the percentage that then looked at biologgers? Because my guess is there's probably not, as you showed, not that many papers.
So if I combine
or maybe wild animal in general, but
Yeah.
whether or not the biologging might actually make up a greater proportion than you might think of those ones.
Yeah. So if I combine the two graphs I showed one after the other, yeah, that would be 1%. 1% would use biologging and consider welfare at the same time. In wild animals, yeah, exactly.
Out of all the wild animal papers?
Sorry?
Out of all the wild animal papers, there's only 1%.
So within the wild animals, so that's a good question. I don't remember, but if I combine all parameters together, that would be only 1%. Only 1% of all the studies you show here combined wild, biologging, and welfare.
I have a question myself. I do work quite a bit with technologies applied to farmed animals. And one of the things that I'm very concerned with is that we try to put a sensor on an animal or outside. And then we do get some indicators, but they are quite meaningless if you think about not knowing the context that's happening. And I think that's even a bigger challenge for wild animals. Have you considered that you need to take into the context that the animal is behaving that way into consideration, and how do you add up other sensors to actually get the context? And then by context, I mean like weather, pressure, whatever, these things that might affect the animal.
Yeah, so as I said, so you can obtain such data through the devices themselves, but you can also use satellite data. So you can really have like very precise data now from satellites, so you can really see how these potential, I insist on the potential, on these, whether these potential markers fluctuate given the environmental conditions that animals are experiencing. But of course, that's very important to have this environmental monitoring at the same time, because as I said, compared to captive animals, you don't know what they experience.
Perfect, thank you. Sorry, Mike. We don't have more. So our next speaker is Kimberly. Hello.
No, it's Kathrin. Sorry.
Well,
I'm
Kathrin. All right,
Yeah.
Kathrin. All right, Kath. So.
Yeah, hi, my name is Kathrin Herrmann. Sorry for the confusion. I'm a veterinarian and diplomat of the European College for Animal Welfare and Behavioral Medicine. I'm specialized in animal welfare science, ethics, and law, and I work at Hopkins University at the Center for Alternatives to Animal Testing, where I direct the Beyond Classical Refinement program. And today I'm here standing in for my co-author, Kimberly Jane. She was supposed to present today, as you saw in the program. And I'm going to talk about some work that we did at CAT in collaboration with Peter UK and Peter US and with Constanza Carvalho from the Institute of Applied Psychology in Lisbon. And we looked at we critically evaluated the forced swim test and its use, especially in depressive disorders research. And so I will start with a brief overview with regards to the forced swim test. My talk will outline where have we come from in terms of the forced swim test, where are we now, and where are we going. And so I will first talk about the scientific and welfare concerns with the forced swim test. Then I will show the local, regional, and national policy revisions that have been in progress because of that. And I will then present a brief study that we did, a literature search, and end with some recommendations for future work. So what is the forced swim test? I'm sure that most of you know that. It is a behavioral test that was initially developed in the 1970s to test antidepressant drugs, and then adopted in an attempt to study depression. And the test involves an animal, usually a rat or a mouse, is placed in an inescapable beaker. There's no safety platform or escape rest platform. And so initially the animal swims and tries to find a way to escape. And then after a while, they stop to swim and they start floating on the surface, and this latency to become immobile, and the duration of floating is then recorded. So historically, what was thought was that floating behavior has been interpreted as as despair. Like it's thought that the animal is in despair, and on that assumption, you say that this is depressive-like behavior when they give up swimming earlier than others. So those are the ones that are depressed. And then this effect was tested or can be reversed as antidepressant drugs. That was the idea behind it. And it's one of the most commonly used tests in an attempt to model depressive-like behavior in animals. And that's why, of course, it's important to see if it's actually doing what it's supposed to be doing. And in recent years, there have been some concerns, or it has been more scrutinized. And they ask if it's really, if this is really depression-like behavior that we are looking at, or and that is the big question. So there are some papers, or have been some papers coming out that say that argue that floating is not a sign of despair, but it is rather a sign of learning that and also of conserving energy, and the animals adapt to the new environment, or it's also seen as a stress coping strategy. So in fact, an accumulation of evidence over the past several years have led to the forced swim test being critiqued on multiple grounds, and in particular, where it is used as a model of depression. For example, when some argue that the forced swim test is a poor model for depression, and it can only be used as a measure of active versus passive behavior. And in terms of the validity, it is said that a short test cannot be used to determine human depression, which develops slowly and not in a test within 15 minutes of swimming, for example. And also an anthropomorphic interpretation of behavior is measured rather than clinical symptoms of depression. That's the other critique. And in terms of being not reproducible, it has been shown that the results vary depending on age, weight, sex, strain handling, social environment, diet, water temperature, and also water depth. And then another problem is then, of course, with translation. It has been shown that differences between rodent and human brain cells and the ways they produce proteins critical to neuropsychiatric function are there, and that's why there's a translatability problem. So the welfare concerns, I think everybody who watched the animal trying to escape this water container sees that there are welfare concerns. And the EU Directive 2010/63 on the protection of animals used for scientific purposes categorizes forced swim and exercise tests with exhaustion as an endpoint as severe procedures. I'm sure that, this is pretty obvious if that this is a problem for the animals' welfare. So the NC3Rs in the UK says this test is highly contentious and under considerable scrutiny and due to its severity and effects on animal welfare. Then it is observed that rodents often defecate during the test, and it is seen as a response triggered by fear. Then, of course, there's the potential possibility of drowning. It also has been documented that animals aspirate water and then later die in the cage. And of course, there are also other, beyond the depression research, this is used in other settings, and they also have welfare concerns. For example, this is used for drug testing, but it's also obviously used as an acute stressor. It is used to induce hyperalgesia, and also it's used along pain sensitivity tests, for example. And so the good news is that given these scientific and welfare concerns, there are many universities and also pharmaceutical companies that then changed their policies, and they said that they don't want to use the forced swim test anymore, or they intend not to use it anymore in the future. So that's a good development. And so we have also seen that the removal of the forced swim test from project applications has happened midway during the review process, which is also encouraging. And as well as in some establishments, they have an actively discouraged list, and the forced swim test was put onto this list. Or the test is being restricted for specific uses. That's also something that can be seen. And there's positive changes on a national level as well, and also regional. For example, in Australia, the New South Wales Parliament enacted a ban on this test, and it also said that national funding councils are not allowed to fund this anymore. And similar developments have been seen or have happened in New Zealand, where actually this test is not used at all anymore. And also it's great to see in the UK, they have a recent policy review of the forced swim test. So what the Home Office did, the Home Office is the equivalent to the competent authorities in the EU member states. And it requested a review from its independent advisory body, and it's called the Animals in Science Committee. And it published the outcomes of the policy decisions in March of this year. And they said that the aim is to completely eliminate all uses of the test in the UK in the near future. And then they said that they want to prohibit the use of the test as a model of depression and for studies of anxiety and its treatment. And also some projects that were already approved without a clear explanation or justification of the use of the test were reviewed again, and some of them were revoked or amended. And so they so they did that. And also for new proposals, it is scrutinized more intensively than before. Oh, okay, I hurry up. I'm talking too slow, sorry. So there is, they also said there's a need for validation for suitable alternatives, including also specific funding calls for that. And also in terms of refinement, where the test is still used, they wanted to refine it. For example, species-specific water temperatures should be amended. And so it's positive to see that science is leading these policy decisions in the UK and elsewhere. So that's really how we are hoping to do things. And so what we wanted to do with our brief literature search is we wanted to see are the institutions still publishing the forced swim test, the EU and UK-based establishments. So we did a search on PubMed and looked for the year 2022, what was published there. And we also wanted to know what's the scientific rationale for using this test. And so we of course, it's just a snapshot of the data, but we just wanted to see also if there's a development away from using this test. And the results, we found 49 publications from EU establishments that used the forced swim test or published it in 2022. And from those, 40 were looking at, they want to measure depressive conditions. Out of them, 7 were not providing a rationale why they were actually using this. And then the others, that used the test did it to stress or to measure sickness behavior. And out of the 40 papers that used the forced swim test in the context of depression modeling, 2 papers acknowledged controversies in the interpretation of the test, and 3 papers noted steps to monitor welfare. Oh God, what did I do now? Sorry. So I just wanted to give a few quotes, what they said in terms of justification. The forced swim test is one of the most popular methods to measure depression-like behavior in rodents. The immobility or floating behavior reflects the behavioral despair often found in depressed human patients. In the forced swim test, we measured the duration of the immobility, which is directly proportional to depression-like behavior. The longer the mouse stays immobile in the test, the higher the depression level. So then I just wanted to, how much time do I? Oh, I do. Okay, I don't have to speed up. Okay, good. Okay, thank you. So also the data we collected so far represents just a snapshot of the time in how the evolving knowledge around the interpretation of the forced swim test is slowly informing scientific literature. If you look at the data in a timeline, it puts it into context in when we first started to see significant publications questioning the interpretation of the forced swim test. That was around 2015. And when it first started to impact local, regional, and national policy, which is from 2018 onwards. So our data, as I said, is from 2022. And so in those 7 years, you could on the one hand see that the test is still used, even though it looks like, you could say it's out of date because the interpretations are questionable. But on the other hand, we could also see that there's a slight reduction in the use of the forced swim test that we could observe. So that's hopeful. And especially because I think there's a paper showing that it takes at least 14 years for new things to be implemented. So maybe we are still in a good time. I don't know how to say that, but it's still hopeful. So in summary, with those concerns about animal welfare and also, of course, questions about its scientific merit, this is very important to note. We want to provide recommendations for stakeholders involved in the funding and the reviewing and publishing of projects that may use the test. And such recommendations can be taken into account when undertaking the harm-benefit analysis, but also when we talk about funding this in the first place. I mean, we heard now about the harm-benefit analysis. It's maybe not an easy tool, but it's all we have to weigh the harms against the benefits at the moment. And as I showed earlier, if this is sometimes even severe, like a severity level of severe, it's really shouldn't be authorized. And also in terms of translatability, obviously. So that's very important. And so in terms of recommendations, enhanced scrutiny for projects that include the forced swim test is our main recommendation. Then the test must not be authorized in depressive disorders research because that's really not fit for purpose. There's enough evidence for that. Then procedures must not be authorized that do not meet the harm-benefit analysis, even if there's no replacement available. And the evolving scientific knowledge must inform project review. And then last but not least, evolving scientific knowledge also must inform policy to safeguard animal welfare, scientific integrity, and also public confidence. And so I just want to leave you with this last statement. We must not rely on anthropomorphic over-interpretation of the behavior of non-human animals when translating it to human clinical conditions. And the forced swim test is just one example of what may be a myriad of other behavioral tests that may restrict progress in the field of mental health. And thanks for your attention. And if you have any questions, let me know.
Thank you very much, Kathrin. So we have a few questions. So we're going to start here in the middle, and then we're going to go all the way up. So
That was pretty fast. I was like,
Hi, Jessie Kate from the University of Guelph. Thank you so much for your talk. I can think of a lot of procedures and tests that we do with lab rodents that are far worse for welfare and have no validity. So I'm so interested to know why you think that the forced swim test has had so much traction with policymakers.
that's a good question, because I think it's just the beginning. But it doesn't make sense, this test. I mean, it's just, I hope I brought this across. I can cite some publications, further publications to read. For example, we did one citation analysis where we looked at who is actually citing those studies where the forced swim test was used, and that's mostly other animal researchers, and never we found no use of people who were doing clinical trials in humans. So that shows that this work doesn't inform human clinical trials, which makes it useless. And yeah, of course, I totally agree. There are so many other tests that are highly questionable, and there's more and more data coming out. But I think because it's such a widely used behavioral test, that's maybe an easy target. It's a low-hanging fruit, I would say. But of course, the tail flick, the tail flick. No, now I can't even remember what the word is. Where they hang the animal on the tail is also awful. I mean, and highly questionable. Not only awful from an animal welfare perspective, but also, I mean, many behavioral tests, as you say, are. But I think it's important to start somewhere and then expand.
we have like 3 questions lining up. So try to make it quick. So it's over here, and then there was someone right here in the middle, and then back there.
Thank you. That was very interesting. So I've used in my research, that was over 15 years ago, the forced swim test. And I think it's really, you've provided a lot of justification as to why it's there. There are a lot of issues with it. One of the reasons why we used it was we're exploring things that we don't know about when we do science. And one of the reasons we used that test is because we wanted to anchor ourselves in the reality of what others had described before us. So that's the reason why there is inertia in the process. And so by extension, sorry, I don't want to make it too long, but what alternatives do you think are preferable?
It really depends what you want to do with this test, but I can point out some papers about, for example, when you talk about depressive disorders, what animal-free alternatives, NAMs, non-animal methods are available, and also what you can do different in clinical trials. I'm happy to send you some papers if, I could do that. So, and what I also hopefully brought across, when it comes to if there's no alternative, if you want to do an animal study, and then I don't think it's justified to still do that, because it's not, if it doesn't lead to a translatable result or even reproducible result, because as we saw, there are strain and age differences, and really there's one paper also where they actually think that the mice observe their other mice, and then they're like, oh, I can do the same thing, and they float it right away. So, I mean, they are smart animals, and so I think it's just not justified to use that. And in some cases, if there's no alternative, then there's no alternative. But I'm happy to send you a list of what I found for alternatives, animal-free alternatives.
Okay, so can you make it very quick, please? And then, sorry.
I have quite a lot to say about that, but I'll try and just make it very quick. It's just really that if there's no scientific rationale for the use of the test and no real justification, no evidence that it's useful in any way, It really doesn't make sense to use it because, I mean, that's obvious to anyone that a mouse swimming or not swimming and just floating, what has that got to do with a depressive state in a person? It makes no sense to me. So doesn't this have implications for the drugs that have already been authorized for the treatment of depressive disorders? So if they've been authorized partly on the basis of this, maybe they don't work.
Yeah, the interesting thing is that there are drugs that are working in humans, but they don't do anything in the forced swim test. I think Prozac, for example, is one of them. So that's my point. Like if you would, I think we are sometimes on the wrong track and that we are misled, and that might lead to drugs not being tested in humans because they didn't do anything or didn't do the right thing in those behavioral tests. But on the other hand, thankfully, some drugs still make it past that and are useful in humans, for example, for example, Prozac. But there are other examples of antidepressant drugs that are not verified in the forced swim test. So yeah, I think we are losing potentially useful drugs also if we continue doing that. And that's why I really hope, I think it looks very promising how many policymakers are changing their policies and how many drug companies are not using that test anymore. I think academia actually, sorry to say, that is sometimes the slowest in adapting or changing their way of doing things, which is a little bit sad, but it's, I guess, because it's, I don't know. Maybe, let's not get into it. We have no time.
Yeah.
But yeah, I can talk about it more later if somebody wants to talk about that.
Right on.
Yeah.
Thanks, Kathrin. Right.
Thank you.
So our next speaker is, at least the one that I have here in my list, is Kaya, or because they're Jasmine, right?
Jasmine.
Yeah. So we are skipping one. Okay. All right.
Yeah.
So yeah, that's you.
Okay. So I'm the last talk of today. So thanks for staying with us. So today I'm going to be talking about a project that I was doing in Glasgow, looking at whether gradual decompression offers a humane alternative compared to CO2 for killing laboratory mice. So I'm not going to go into this in too much detail because we've heard about it throughout the last 2 days, but when we're talking about laboratory rodents, we're talking about a large number every single year. So a really conservative global estimate is that there are around 192.1 million rodents used every single year for biomedical research. Now, unfortunately, often a prerequisite to their use is that they've got to be humanely killed upon completion of the scientific work, either because they've reached the end of the scientific study or if they're surplus to requirements. And therefore, I really wanted to highlight that it does therefore represent the most widely used procedure. Now, in the UK, this is regulated by Schedule 1 of the Animal Scientific Procedures Act, and obviously in the EU, it's under Annex 4 of 2010-63, and they permit various different methods for killing laboratory rodents, and these include overdose of an anaesthetic, exposure to carbon dioxide gas, cervical dislocation, and striking the cranium or blunt force trauma. So I'm sure you're all probably very aware of the kind of negative attention that exposing rodents to CO2 has gained in the past 20 years, largely due to its ability to induce negative sensations such as panic, fear, anxiety, and also pain when exposed to high concentrations. And therefore, that's why we were interested in looking at an alternative. So for this particular project, we were interested in the potential of gradual decompression because it too could also provide a high throughput approach and also a non-contact approach for human operators. So there's been growing interest largely due to the work done by Dorothy McKeegan in poultry, and essentially hypobaric hypoxia results due to low atmospheric pressure conditions, whereby as the atmospheric pressure falls, you get a reduction in the partial pressure and therefore the availability of oxygen. And as such, it simulates a process equivalent to ascending to a high altitude. And the reason it's gained significant traction is because we know in humans exposed to hypobaric hypoxia, such as those in aviation training, for example, that it's considered insidious people often don't know they're becoming dangerously hypoxic before they have a very short period of time to act, because they become cognitively impaired before losing consciousness. So I'm sure we all can appreciate it would be great if we could ask animals how they feel, but we obviously have to think about how we can effectively assess their welfare, which does get a little bit more difficult when considering doing so during the killing process. So a lot of methods aren't instantaneous, including CO2, and therefore a really important thing to derive is what happens to that animal before it loses consciousness. And therefore, we've got to look for the time that it takes them to lose consciousness and also their potential to experience negative sensations during that time. And scientifically, we can also look at employing things such as drug interventions in order to give us an idea of whether any behaviours that they may be experiencing or showing are likely mediated through pain and or anxiety, for example. So for this project, we took a really systematic iterative approach with the overarching aim of evaluating the potential of hypobaric hypoxia to provide a high welfare method for killing laboratory rodents. So the first step was very much proof of principle. We didn't want to expose conscious animals to a method that we A, didn't know was feasible, and also B, it wasn't associated with gross pathological damage, and therefore, due to the fact that these animals were terminally anaesthetized, I'm not going to talk about the data in this talk, but it is published if you are interested. So following that, we did proceed on to conscious animals, and we performed a behavioural and pharmacological trial. And then after that, we looked at a more appropriate measure of loss of consciousness through looking at their brain activity using EEG approaches. So for the behavioural experiment, we employed a slow overall rate of decompression, and this was very much in line with the human literature, and that was an ascension equivalent of 40 meters per second. So we know from humans that they don't experience any barotrauma when they're exposed to such a rate. We compared this to CO2 employed by good practice. So in the UK, that's at a 20% chamber volume per minute, and that's filled from the top. And we also had a sham treatment group as well, where the animals was just placed in that internal perspex chamber with vet bed, and obviously nothing happened. And then within each of those, mice either received an analgesic, an anxiolytic, or a saline injection. And it was all filmed for, obviously, comprehensive behavioural assessment. So I did want to show kind of the differences between decompression and CO2 in terms of the mechanisms. So you can clearly see from our decompression profile here that as the atmospheric pressure falls, if you work out the equivalent oxygen as if you were at sea level, you can see this relatively linear reduction. This is very different to what's happening with CO2. Instead, you obviously get a reduction in oxygen due to an influx of an alternative gas. The other thing I wanted to kind of highlight here is the differences in the mean time the animals take to lose posture, which is often taken as a behavioural proxy for loss of consciousness. So you can see that due to the more gradual nature of decompression, the mice are losing posture significantly later than CO2, and therefore we've always got to remember that when we're looking at interpreting these two methods. So in the essence of time, I've kind of tried to summarize the key findings from this particular experiment. And essentially, we found that despite the mice taking longer to lose posture with decompression, they are still performing normal species typical behaviors. So they're spending time exploring their surroundings, and they're also performing grooming related behavior as well. And therefore, for me, the fact that they're performing such normal behaviors during this longer induction is really encouraging. We also found no effects of our drug interventions on the expression of any of these behaviors either. In terms of behaviours that tell us about something potentially concerning, we did see that mice performed ear scratches and head flicks. However, this is where the beauty of having a sham group comes in, because we actually found that the sham mice were also ear scratching and head flicking. And statistically, there was therefore no difference in the likelihood of them ear scratching if they were undergoing decompression. And also we had no effects of our pain relief or anti-anxiety medication on their expression either. However, what we found for CO2 is very much in line with probably what we all would expect, given all the previous research on this topic. And we found that mice were more likely to gasp compared to decompression. And although not statistically significant, given the few mice that actually did these vertical escape attempts, we found that mice were obviously performing more escape attempts with CO2. And you'll also note that of those mice in CO2, they're actually all saline treated mice as well. So moving on to the kind of EEG experiments, I would love to have put head caps on the mice, but unfortunately they're not gonna have that. So we surgically implanted these wireless EEG transmitters, and essentially what we wanted to do with this experiment was to better appropriate measure or physiological measure of loss of consciousness. And this is really important when considering that welfare assessment. We want to determine the time that it takes them to lose consciousness, and that's our period of concern for animal welfare. However, like with all methods, it's not without limitation. It doesn't give you an exact point at which think of it less like an on-off light switch and more of a dimmer switch. In this particular experiment as well, we compared CO2 filled from the top and also CO2 filled from the bottom. Even though the last experiment that I've just described was top fill, what we found is we did a recent survey and found that despite it being considered best practice, a lot of places across the UK are actually still bottom filling. So we wanted to determine whether we got any behavioural effects between our CO2 treatments, but also obviously anything in terms of their brain activity. So we obtained 22 recordings from mice undergoing one of 3 terminal treatments. And I'm sure you can all appreciate that irrespective of the terminal treatment, there's a really consistent change in the visual appearance of this EEG. So at baseline, when the animals are obviously moving freely in that inner perspex, we've got this high frequency signal, which upon application of the terminal treatment becomes more slow wave, which in laboratory mice is considered less than 4 hertz. So that would be at an anesthetic plane. And then ultimately, as the terminal treatment progresses, the signal becomes isoelectric, which is essentially a really conservative measure of brain death because that purely represents electrical noise from a non-biological origin. So I'm not going to try and over-explain this too much, but essentially it's a really visual representation of what's happening in the brain during our 3 terminal treatments. So we've got a frequency on the Y and time on the X. Our dashed horizontal line at 4 hertz is that threshold in which you would consider an animal, or in this case, a laboratory mouse, unconscious. The red arrows denote the mean time for them to lose posture behaviorally. And essentially, the more yellow or red means the greater power in that frequency range. So essentially, it's showing you the dominant frequency range across time. So you can clearly see for CO2 that we have activity that is dominant in the alpha range initially that transitions through the lower frequencies. And it doesn't actually hit below or become greatest or dominant with CO2 until after the animals have lost posture, which I think is really, really important. With decompression, you can clearly see we've got a completely different kind of pattern of activity. Instead of that slow progression through the lower frequencies, we have this stepwise transition straight to the theta delta boundary. And that's happening substantially before the animals lose posture. And therefore, this to us indicates early cognitive impairment and that transition towards unconsciousness, which is occurring substantially before loss of posture. In terms of what they're doing during this time behaviourally, what we wanted to look at is obviously that time course and relate that to our EEG activity. So here you can see that we're obviously getting a range of behaviours occurring before that transition to slow wave activity. But we are seeing, once the brain is in that slow wave range, we are seeing things such as respiratory related behaviours occurring after that. and therefore the indication that the animals could be cognitively impaired at this point and have some impaired consciousness. They're not fully unconscious, but they are likely to be impaired in some way. A lot of the respiratory related behaviours are happening during that time and therefore could indicate reduced sensations owing to respiratory distress such as gasping. Now, this is very different to what's happening with CO2. Again, the red arrows there denote the time from our time frequency spectrograms. And again, what you can see here is that we've got a whole range of behaviours. However, respiratory related behaviours such as gasping, then becoming ataxic, and also loss of posture and escape attempts are all happening before the brain is in that slow wave range. We also wanted to look at counts of behaviours, given that that can provide meaningful information as well about the animal's likely experience. And in accordance with our previous behavioural experiment, we found, again, greater gasping with mice exposed to CO2, more escape attempts, and once again, decompression mice were performing relatively normal despite this kind of longer induction phase. However, in this particular study, given that we didn't have a sham group in this particular experiment, we did therefore find that mice undergoing decompression were more likely to ear scratch. However, combined with our findings from our behavioural trial at this stage, we are concluding it's unlikely to reflect pain and/or anxiety. Finally, it was kind of in relation to the CO2 methods, this was kind of quite a striking and surprising finding for me, even though it's common sense. Although the top and bottom fill CO2 treatments were at the same flow rate, so the gas was pumped in at exactly the same flow rate, we've obviously found this striking difference between the treatments in terms of their concentration at the level of the animal. And that's obviously because CO2 is heavier than air. So if you pump in the CO2 at the top, you get a more even dissipation. Whereas if you pump it in at the bottom, you get a really kind of big accumulation at the bottom, which happens to be at the level of the animal. And therefore, I think it's really important that the concentration doesn't purely rely on the fill rate. And perhaps we should also be specifying methods and giving more details as to what we should be doing, because there is the potential that bottom fill mice are exposed to higher concentrations, which are also capable of eliciting pain. So the key findings from our neurophysiological experiment is that it's clear from the spectrogram that brain activity differs during decompression and CO2. For decompression, we have this dominance of slow wave activity occurring substantially before loss of posture, but also occurrence of these respiratory behaviours, and therefore could suggest reduced negative sensations owing to respiratory distress. With CO2, this is obviously very different. Slow wave activity is occurring after loss of posture, and also respiratory related behaviours, and therefore they could be experiencing these negative sensations of respiratory distress. I think for me, this was really kind of striking as well in terms of the animals losing posture but not being fully unconscious, because a lot of institutions rely on the animal losing posture before they'll turn the gas concentration up. And therefore what this shows is that there's a chance that if the animal's not fully unconscious at that point, you're then putting a higher gas concentration in, and they therefore could experience pain. And of course, with the bottom fill, this is kind of further exacerbated, whereby it builds up at the animal level and again could elicit greater pain. So I hope you'll all agree that at this stage, the data shows that gradual decompression could offer major refinement for the way that we kill millions of laboratory mice worldwide, and as such could have implications for a whole range of policies and national guidance. So at the moment, we are trying to further assess its feasibility in mice. So I'm really keen, aside from my current postdoc project, to kind of really further explore this, in particular, try and get to the bottom of the ear scratching a little bit. But we're also exploring its potential in rats. So we did the first proof of principle experiment in rats. It was effective in inducing a non-recovery state and with minimal gross pathological damage, and therefore Sophia Bullock's going to be doing her PhD at Newcastle, looking at whether it offers promise for rats too. And finally, I'm also working on a UFAW funded project assessing the effectiveness of cervical dislocation, because a lot of recent data has suggested that because of the negative attention that CO2 is getting, a lot of people are therefore doing cervical dislocation. But from moving around universities and different institutions, I've kind of found that everyone does it slightly different. And then again, there's no kind of guidance on what you should be doing and how you should be doing it. So that's something I'm going to be exploring, and UFAW kindly funded that little project. So of course, I'd like to acknowledge all the project team. It was a hugely collaborative effort, including all the staff at Glasgow. We had a lot of support, and yeah, so thank you very much.
Thank you. Do we have any questions for Jasmine? So we have one over here.
Hello, Angela Bergschmidt from Thünen Institute in Germany. Would you know if there are any similar experiments having been carried out for pigs for gradual decompression? Because they face the same, I think, aversive reaction against.
Yeah, so as I joined Glasgow for this particular project, Dorothy McKeegan was PI on a DEFRA-funded project to look at the potential of decompression for pigs. I think there's the initial few papers are out already, and there's a couple still to come for that. But essentially, it doesn't look like an alternative for pigs compared to CO2, because they were seeing rupture of the eardrums. And also, I think because the beauty with sorry for the phrasing, the beauty with the kind of this work is that I don't have to obviously, I'm not constrained by time as much as in a commercial slaughter setting. So the rates that they were having to kind of go to with the pigs were higher than what we were doing here. So it was kind of one of those balances between, well, we can't go any slower because it'll compromise the meat quality. So yeah, it was concluded that it just wasn't a humane alternative for pigs, unfortunately.
Any more? Oh, Mike.
Thanks for a great talk, Jasmine. Is the slow wave EEG is sort of indicating a sleep-like status, or what's the sort of gradual loss of consciousness, and why is there a delay before postural collapse?
It's really strange and something I can't really answer. So the animals aren't asleep because they're still moving around at that point. But subjectively, when I was watching them, it's kind of like the lights are on, but no one's home. Because they just don't seem to be doing motivated behavior at that point. It's more just kind of like paddling. So they're not asleep, but they're obviously not fully conscious either. So it's this transitory phase. So I've been trying to delve a bit more into the human kind of literature on people exposed to this for obviously like airline training. And people do report that cognitive impairment, but I haven't kind of found anybody that's done an EEG at the same time, because I would love to have seen whether we're seeing similar transitions. Because there is a really good documentary by BBC a few years ago where Michael Patillo underwent decompression, and they're asking him to do really simple tasks that you would ask a child to do, and he just cannot do it. So whether it's reflecting something similar, I don't know. It's kind of hard to say at this point. And that's kind of why I would like to explore it a little bit more. So it'd be quite nice to do some more like operant type things. Then we could get at what are they capable of at that point.
Any more questions? I guess nope. So yeah, that's it for today.
Okay, everyone, thank you very much for coming back. I'm really impressed how many of you came back at the very end of this meeting. So thank you for that. I'm just going to wrap up, and the first thing to do to wrap up is to just announce the results of our poster competition. So various of us have been sneaking around the poster exhibition, working out which posters we thought were the best ones. So thanks to the guys who've been doing that as well. So I'm just going to quickly announce We had two that were highly commended and one winner. So the two highly commended ones were Anne Clay and colleagues from the Wild Animal Initiative, with the poster on addressing gaps in our knowledge for measuring wild animal welfare, and Marissa Estevez Montero and colleagues from the University of Porto for their work on enhancing diabetes mellitus research through the application of the 3 R's principles, which is something close to UFAW's heart. So congratulations to both of those two authors, if they're here. And the winner, I'm not going to do one of those terrible delays like they do on TV, is Andrea Pisedu and colleagues. For this super clear and really visually engaging poster on their Delphi consultation on indicators of parrot welfare, which is another topic close to UFAW's heart. So Andrea, I don't know if you're here, but we will send you an email and offer you one of the books from our UFAW book series as a prize. So congratulations, he's over there. So. Fantastic. So really, I think the main thing for me to do now is say a few thank yous and just close the meeting. I won't try and be really clever and remember all the highlights of the meeting because there are basically too many. And as you've seen, we have pretty much the most eclectic conference schedule you can imagine. But there's been some absolutely amazing science on the broadest range of topics. And then we've just been in a really fascinating workshop looking at what you might do with your careers if you don't work directly in animal welfare academia, which was also, I think, really, really helpful, both for the people in there and for me as well, thinking about how we might support people to be effective in animal welfare. So thank you to everyone for coming. I'd like you to, as well, to tell us how it was for you so that we can improve these meetings in future. So do tell us what you thought worked well, what didn't work well, and most importantly, what you'd like us to do at the next meetings that we run. So I'm going to show you a couple of links for surveys and QR codes. We'll email these out to you as well. But first of all, this is just the one for the people who are here in person. Hopefully we'll be putting a link on the chat for the Zoom people, but if you're here in person, scan the QR code or follow the link and tell us in just a few short questions what you thought we did well and what went badly. And we do already know about the technical issues with Zoom, but feel free to tell us again. I'll give you a minute or two to fill that in. And then I'll just quickly, this is the one for the folks online, if anyone wants to scan the one online and tell us how that went for you. And apologies to those folks, because we've definitely had a couple of technical glitches with Zoom, but hopefully it didn't spoil your experience. And we will have recorded all the talks. So if there are any issues, you can watch the talks back at a later stage when we get them up on YouTube. Okay. And I'd just really like to ask you to stay in touch with us at UFAW. If you sign up for our new e-newsletters, which you'll start getting soon, using that link or that QR code, we'll let you know about what we're doing next. I guess we'll be doing a conference probably online next year like this. And then in 2026, it's going to be in London, back in the UK. Not because we want to do one close to home necessarily. I always love coming to exotic places like that. But UFAW was founded as the University of London Animal Welfare Society in 1926. So it'll be our centenary in 2026. So we figured we better go back to where we started out. So I'd be hoping to see lots of you in London in a couple of years' time. And now's your chance to tell us what you want in that meeting. And we'll try and put the program together to give you what you most want from these meetings. Thanks to our sponsors, Charles River, again, that's really helpful to us to have sponsorship to make these meetings possible and to allow us to make them accessible to people who would struggle to afford to come without support. And most importantly, thank you to all the people who made this meeting possible, which has very little to do with me and all to do with the folk at UFAW who actually put all the work into getting this meeting together. most importantly, Steve Wickens, who you've heard plenty from over the last couple of days, but has been working on this meeting for, God knows, probably more than a year, putting absolutely everything in place and doing all the really hard work, making sure that it runs properly. Theatre, who you've seen today, Carly, who is our comms person, who's back in the UK with COVID and has been doing all the live tweeting despite having COVID and not being able to join us. So huge thanks, especially to her. Ailey, Steve from the Journal, and the folk back at the office, Sam, Tina and Jane, who've done all the admin. And really importantly, our hosts at ICBAS and the University of Porto, and particularly Paolo and Anna, who've done all the local organising. So yeah, huge round of applause to everyone who's helped out, also our student helpers and everyone who's organised the catering and all that sort of stuff. And most of all, thank you to you guys for coming. Finally, I can't ever end this without saying, give us your money. We are still a charity and we're still funded solely by your donations and legacies, and we do use them for really good stuff, like, you know, running these meetings and making sure that people from the Global South can attend, at least online, and those kind of things, and funding as much research as we can, including quite a lot of projects that you've heard about over the last couple of days. So I will end with thank you very much, but please at least consider joining UFAW if you aren't already a member. Ailey's still here, so you've still got a chance to sign up. But other than that, thank you so much for coming. It's been a fantastic couple of days, and I hope you have some time to enjoy Porto now. And oh yes, and thank you to the speakers as well, Steve says, if I didn't say that. Good point. And the session chairs. And the host. Yeah. We get there. We're going home now, so the weather will improve in Porto, I hope. So I hope you've got time to enjoy it. And thank you to absolutely everyone who's made this meeting a success. Thank you very much.